In David Sinclair's newest paper, his team shows that cocktails of small molecules have the potential to reverse aging. In this video, we'll highlight the important findings in Sinclair's newest paper and whether this study moves us closer to turning back the aging clock. Let's dive in. In this study, Sinclair's team first created a system to help screen for small molecules that could potentially rejuvenate cells. The system they chose, called nucleocytoplasmic compartmentalization, or NCC, is based on the known phenomenon where the cell nucleus, the compartment where most of the DNA is stored, deteriorates with aging, allowing proteins and molecules to leak through. The NCC system enables Sinclair's team to test the effect on cell aging and many small molecule combinations efficiently. Curiously enough, they chose to do the bulk of their tests on rodent senescent cells as a representation of aged cells. If you don't know what senescent cells are, they're cells that stop multiplying, hang around, and can release inflammatory signals, sort of like some YouTube commenters. They then showed that markers of nuclear permeability can be reduced with partial Yamanaka factor or OSK treatment, which is a form of gene therapy that has been shown to reverse cellular aging through a process called epigenetic reprogramming. OSK treatment partially restored the degree to which genes were expressed, or in other words, turned on or off. These findings are in line with the previous discovery that Yamanaka factors could convert adult cells into younger cells, or induced pluripotent stem cells, iPSC. Now that they had a high throughput system where they could drive aging forward and in reverse, Sinclair's team moved on to searching for small molecule cocktails that could rejuvenate cells through a process called chemical reprogramming. They then tested over 80 cocktails in total with their NCC system, which led them to focus on the 6 most effective cocktails, 3 for human and 3 for mouse cells. These 6 cocktails contain molecules such as valproic acid, sodium butyrate, and alpha-ketoglutarate. Looking at gene expression, they found that the 4-day treatment with these cocktails only partially rejuvenated the cells, which is a good thing since we don't want cells to fully revert to stem or stem-like cells, and generally affected the same group of genes, suggesting that they're working through similar mechanisms. Out of the 6 cocktails, cocktail number 1 was the one that rejuvenated the cells the most, which was shown using the NCC system in mouse cells after a 4-day treatment. Unfortunately, the effects on human cells were not as potent compared to the mouse cells. Using a different aging biomarker, a transcriptomic clock, which tracks gene expression in aging, they then show that these 6 cocktails partially revert the age of rodent senescent cells. Sinclair's team performed correlation analysis with data from the cocktail-treated cells, which found that the transcriptomic clocks were more similar to those from young cells compared to old cells in mouse, rat, and human organs. Looking closer at what the genes affected by these small molecules do, they found some are linked to mitochondria, hypoxia, and inflammation pathways. Since epigenetic and chemical reprogramming can cause cancer formation, especially a type of cancer called teratomas, the Sinclair team took an initial look but did not detect changes in markers of cancer, such as increased levels of the pro-tumorigenic gene NANOG after treatment with cocktails. While not the first to show that chemical cocktails can turn back the clock in cells, it's encouraging to see this study from Sinclair's team support that rejuvenation is possible, at least at the cellular level. Chemical cocktails have advantages over gene therapy, such as Yamanaka factors, since they are cheaper to make and potentially safer, which could accelerate the timeline for regenerative medicine development. Though, the trillion-dollar questions remain. Can we find a combination of small chemicals to rejuvenate organs and ideally organisms? And what treatment regimens or complementary chemicals will be needed to significantly decrease or ideally eliminate the risk of cancer formation from chemical reprogramming and known side effects from these drugs, such as valproic acid? Sinclair claims that one of his companies, Life Biosciences, successfully used Yamanaka factors to rejuvenate vision in monkeys in an age-related vision loss model, and that human trials will soon occur. But this evidence has not been published or replicated, causing many scientists in the field to harshly criticize this paper and some of Sinclair's seemingly hyped messages to the public. Before there's substantial proof that Yamanaka factors or chemical cocktails can turn back the clock in animals and safely, it would be best for Sinclair's career and the longevity field for him to back up his claims with peer-reviewed data. Nevertheless, the advancements in technologies and our understanding of molecular biology has me optimistic that our genome and epigenome hold the key to extending human lifespan and that we're getting closer to major anti-aging breakthroughs.